presentation, I'll compare two prominent aDNA sequencing techniques, the 1240K capture assay and Twist Biosciences DNA sequencing technology. So let's embark on the journey to discover which technique stands out. Before diving into the comparison of the two DNA sequencing technologies, it's essential to understand the fundamental concepts of DNA sequencing, especially in the context of ancient DNA. So let's have a look at DNA first. DNA, or deoxyribonucleic acid, is the molecule that carries genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. Located in the cell nucleus DNA, it is organized into chromosome pairs and structured as a double helix shape. The double helix is composed of four nucleotide bases: A for adenine, T for thymine, C for cytosine, G for guanine. These base pairs, specifically A with T and C with G, form the rungs of the helix. In understanding of DNA, it is also crucial to define two key terms: a gene, which is a segment of DNA that contains coding for a protein or functional product, and a genome, which is the complete set of genes or genetic material present in a cell or organism. Next, aDNA, or ancient DNA. It refers to genetical material extracted from the remains of organisms that lived in the past. This DNA is typically obtained from bones, teeth, hair, and other preserved tissues. Over time, aDNA degrades due to chemical, environmental, and biological factors, resulting in fragmented and chemically altered sequences. Moving on to aDNA sequencing. This is the process of determining the sequence of aDNA fragments, and it involves sophisticated techniques. Bioinformatics tools are very crucial in analyzing and comparing aDNA sequences to modern genomes. aDNA sequencing has been pivotal in studies of ancient populations and biodiversity, providing insights into human migration, domestication of plants and animals, and the genetic basis of ancient diseases. Moving on, the twist capture assay. This is the capture assay deployed by Twist Bioscience, which is a DNA sequencing company, and it employs advanced methods for selectively isolating and sequencing specific regions of the genome. This technique targets approximately 1.4 million single nucleotide polymorphisms, also known as SNP, across the human genome. The process involves creating custom DNA libraries, performing target enrichment, and achieving high precision in sequencing. While this method is widely used in cancer research, genetic disease studies, and microbial genomics, its adaptability makes it suitable for a wide range of genomic research applications as well. Moving on to the 1240K capture assay. This is specifically designed to target and sequence about 1.24 million SNPs across the human genome, and this method is particularly advantageous in studies where the quality and quantity of DNA are very limited, such as ancient DNA research. The 1240K capture assay is extensively used in studies of human evolution, population structure, and migration patterns, and also genetic diversity, and it makes it a very powerful tool in understanding our genetic history. Moving on. So, why are we doing all this? What's the motivation? So, DNA sequencing is a very challenging and rewarding task at the same time. Conducting research in ancient DNA, we face a lot of challenges that include contamination, where modern DNA can mix with ancient samples, making the analysis very difficult. Additionally, the DNA we work with is often fragmented and degraded due to the passage of time. These factors, combined with the high costs and time-consuming nature of sequencing and analysis, make this field particularly very demanding. And now if we look at the rewards side of the task, the rewards are pretty significant as well. Understanding human evolution helps us piece together our ancestry and migration patterns. Investing genetic diversity offers insights into how populations have adapted over time. Furthermore, ancient DNA can reveal the spread of diseases in historical populations and how they adapted their lifestyle to survive. Now, if we think about the objective of our research project, uh, it's to determine which ancient DNA sequencing technology performs better. Is it the 1240K or the Twist Biosciences DNA sequencing technology? Our evaluation criteria will include the accuracy of determining the sequence, ensuring the reconstructed genetic information is as close to the original as possible, the ability to detect variations, which is very crucial for understanding the genetic diversity and evolutionary changes. Then, next criteria is the contamination estimates in order to evaluate how well each method can distinguish between ancient and modern DNA. And finally, sample properties, assessing how different techniques handle DNA quality and quantity variations in ancient sample. Moving on, the data set. So, in order to provide a clear understanding of the data set we worked with, let's have a look at these key details. Firstly, the output format of the dataset is eager, which is a standard format used in ancient DNA research. We have a total of 50 observations, each representing a unique sample, and the dataset includes 47 variables, out of which 13 are particularly important for our project. Among these 13, we have identified two variables as our predictors: source, which indicates the sequencing technique used, and number of input reads, which refers to the input DNA fragments, are the two predictor variables. We also have 10 response variables. These response variables will help us to evaluate the performance of the sequencing techniques in various aspects. We will find the details of these response variables in the appendix, where the definition of all these 10 response variables are given here. Jumping back to the data set, we also have one random effect variable. Um, it is the sample ID. This variable accounts for the variability between different samples, ensuring our models can accurately reflect the differences in DNA sequencing results from different sources. Moving on, the methods. So the first method that we used is the simple linear regression model. This is used for predicting a continuous dependent variable based on one or more predictor variables. It helps to understand the linear relationship between two variables. We also used logistic regression. This model is used when the dependent variable is binary, and it estimates the probability of a binary outcome based on the predictor variables. We also used simple mixed effect model. This model is ideal for data with hierarchical or grouped structure, where both fixed and random effects are needed. It accounts for variability within and between groups. And finally, we used beta regression model. This model is used when the dependent variable is a proportion or percentage within the open interval 0 to 1. It is especially useful when modeling rates and proportions. Um, Moving on, in the part A of our project, we focused on deploying appropriate model based on the type of response variable. For variables such as covered SNPs on 1240K and number of mapped reads over 30 base points, these are continuous variables, and so we used linear regression models. And when dealing with binary response variables like percent endogenous DNA over 30 base points or proportion of duplicate rates, logistic regression models were deployed. And then in part B, we extend our analysis using models that cater to more complex variable types. So for hierarchical data, we used mixed effect models, and for variables representing percentage or proportions, we deployed beta regression models. By selecting the right models, our idea was to accurately capture and analyze the nuances in our data. Moving on. So if we look at the results, as we can see in this table, For the 7 out of 10 variables we analyzed in this project, supported that twist is the better performing model than 1240K. For example, if we look at covered snips on 1240K, the coefficient 1.1725 represents the difference in the response variable between the two levels of the source variable twist and 1240K. After accounting for the effect of the normalized number of input reads, it suggests that when the source is twist, the response variable is higher by 1.1725 compared to when the source is 1240K. This coefficient is statistically significant as p-value is less than 0.001, indicating that the choice of sequencing technology significantly affects the covered SNPs. Similarly, the coefficients of the six other response variables, as shown in this table, also can be interpreted this way to establish an indication of the conclusion that twist might just be the method that can outperform 1240K. Moving on. Next, we used mixed effect models for the same six response variables for which we used simple linear regression model in part A. Mixed effects models test the random effect of a predictor. Until now, we have only tested the fixed effects of the predictors, but there can be different samples coming from different individuals. So, does an ADNA sample coming out of different individual have an impact on the sequencing technology's performance? To answer that question, we do need mixed effect models. In this scatter plot that we can see in the slide, we have used different colors for different samples, and this shows that there is some sort of random impact, as we can see here that for some of them the line is upward and for others it's downward. Next, to test the random effect of this sample, we used R ANOVA. It told us if the random effect of the samples are significant or just the fixed effects would be better. This analysis can be justified as the response variable was scaled into values between 0 and 1, but not inclusive. Moving on. For the remaining four response variables, we used beta regression model. Here's the visualization of one of the response variables out of those four. The results are relatively better compared to the plain vanilla logistic regression. So if we look at overall, in terms of significance, source has significance across 9 out of 10 response variables. Number of input reads also varies. And if we look at the better technique column here at the rightmost part, for 8 out of 10 variables, it clearly says twist is the better performing model. Uh, and for another one, upon further analysis, we can say twist is better for that as well. So which DNS, so if we have to conclude now which DNS sequencing technique performs better, the answer would be twist, because it outperforms 1240K in 9 out of 10 performance parameters, and it targets 160,000 more SNPs compared to 1240K method. So our next course of action will be test of uniformity. So do any of the sequencing techniques hit the SNPs uniformly, or some technique tend to hit certain SNPs more frequently? To answer that question, we need to test uniformity. We can do that with multiple correspondence analysis. The expectation is that the technique that reaches closest to the uniform distribution will outperform the other one in this performance metric. That's all for today. Thank you.